In this video, we will be highlighting best practices for thawing, culturing, and banking HEK 293 suspension-adapted cells, including Gibco Expi293F cells, Gibco viral production cells, and Gibco viral production cells 2.0. While this video focuses on 293 cells, the best practices provided here are generally applicable to other cell types as well. In a biological safety cabinet, fill a 125 milliliter vented, non-baffled, sterile shake flask with 30 milliliters of pre-warmed culture medium. Obtain a vial of cells from vapor phase liquid nitrogen storage and thaw by placing the vial into a 37°C water bath until only a small amount of ice remains. Do not submerge the vial in the water. Decontaminate the vial with 70% ethanol. Invert the cell vial to mix. Transfer 50 microliters of cell suspension to a cell counting cup containing 450 microliters of PBS for viability and viable cell density determination by trypan blue dye exclusion assay. This PBS dilution step is recommended to minimize the potential for trypan blue aggregation with components common to cell culture medium, which can lead to false dead cell counts. Transfer the remaining cell volume dropwise to the shake flask containing pre-warmed culture medium. Place the flask onto an orbital shaker in a CO2 incubator at 37°C with greater than 80% relative humidity. Perform the first subculture when cell viability is greater than or equal to 90% and viable cell density reaches greater than 1 million viable cells per milliliter, typically 3 to 4 days post-thaw. For routine cell culture maintenance, subculture cells upon reaching the specified viable cell density noted in the user manual. Repeat these steps to maintain and expand cells for banking. It is critical that chemical compatibility be maintained throughout the cell banking process to ensure cell health. For all steps involving contact with DMSO, it is strongly recommended to utilize polypropylene, glass, or other DMSO-compatible materials to minimize risk of cell toxicity. A chemical compatibility guide can be found at thermofisher.com/chemical compatibility. It is recommended to use cells between 3 and 5 passages after thaw for banking and conditioned medium. To harvest conditioned medium, remove an extra flask of cells from the incubator and transfer contents into a sterile polypropylene centrifuge bottle. Centrifuge the cells at 300 times G for 10 minutes at 2 to 8°C. Decant the supernatant into a new sterile polypropylene bottle without disturbing the cell pellet. The decanted supernatant will be used as the conditioned medium. Chill the conditioned medium at 2 to 8°C for a minimum of 2 hours. In a sterile polypropylene bottle, prepare the required amount of 15% DMSO in cold, fresh culture medium using a glass serological pipette. This represents a 2x freeze medium. Keep cold until use. Transfer the required number of cells into a sterile polypropylene centrifuge bottle. Centrifuge the cells at 200 times G for 10 minutes at 2 to 8°C. Decant the supernatant without disturbing the cell pellet. Flick the bottom of the centrifuge bottle to loosen the cell pellet. Resuspend the cell pellet by addition of conditioned medium using a wide-bore pipette and gentle trituration. Add additional conditioned medium to the centrifuge bottle to obtain a 2x cell stock. For example, if banking at 10 million cells per milliliter, prepare a 2x cell stock at 20 million cells per milliliter. Swirl and keep cold until use. The next steps should be performed as quickly as possible to limit DMSO exposure time. Transfer the calculated volume of 2x freeze medium to the bottle containing the 2x cell stock. Swirl the bottle to ensure a homogeneous mixture and keep cold. Immediately dispense 1.1 milliliter aliquots into labeled polypropylene cryovials, swirling the cell suspension before each refill of the pipette. Keep the cell suspension cold during the entire aliquoting process. Try to keep DMSO exposure time under 60 minutes before initiating the freezing process. Transfer the vials to isopropanol-containing cryo-freezing chambers and store at -80 degrees Celsius for 24 to 48 hours, and then to final storage in vapor phase liquid nitrogen. This freezing regimen approximates a 1 degree Celsius per minute cooling to -80 degrees Celsius, followed by a 2 degree per minute cooling to final storage temperature. These cooling conditions may be utilized as a basis for controlled rate freezing.